Hi, thanks for having me. My name is Jessie deHaan, and today I'm going to be talking about the work that I've been doing during my master's in Dr. Miriam H's lab at Brock University. The topic of my presentation today is the effect of developmental temperature on Ceratina calcarata, offspring body size and physiology. So insects come in many sizes, but in self-feeding insects, body size is primarily determined by how much food they eat as larvae. A mother insect lays her eggs on or near a food source. The eggs hatch and the larvae consume as much as they can before they pupate. If there's abundant food, the offspring are large, and if food is scarce, the offspring are small. Differences in body size among individuals of the same species is a form of phenotypic plasticity. The continuous range of body sizes observed in insects is not due to genetics, is due to environmental influences such as temperature or resource availability. Some insects are smaller when they develop at higher temperatures, and this is known as the temperature size rule. Temperature size rule can be explained simply because insect metabolic and developmental rates increase with increasing temperature. They spend less time feeding, they consume less food, and they grow to be a smaller body size. The temperature size rule is applicable to insects that feed themselves, also known as self-feeders or ad libitum feeders. But what about insects whose mothers dictate how much food they get? In mass provisioning insects like bees, the mother dictates how much food their offspring get. Their body size is, again, tied to how much food they eat as larvae, but the difference between mass provisioners and ad libitum feeders is who's acquiring the food. This means there's no clear connection between temperature and offspring body size. If the temperature size rule does apply to mass provisioners, in theory, a mother bee could simply bypass the effects of the temperature size rule by providing more food to her offspring if they're developing at a higher temperature to compensate for the higher metabolic and developmental rates. If developmental temperature does affect offspring body size in mass provisioners, the mechanism is different. For instance, if maternal input is the same, significant metabolic differences could be causing body size differences in bees. A few studies have linked offspring body size to temperature in bees. They're listed here in two species of Osmia, Osmia ribiflorus and Osmia bicornis. Offspring from warm nests were smaller or lighter than their cool or control nest counterparts. But these studies didn't examine maternal input, and the authors hypothesized that the body size differences that they found were caused by metabolic rate differences. So I use Ceratina calcarata as the study species for my master's research. They're a common backyard bee found in southern Ontario. They're very easy to study, even during a pandemic. In nature, they nest in raspberry, sumac, and teasel twigs. And the nesting location or substrate chosen by the mother affects the temperature that her offspring experience during development. So we know that C. calcarata follows Bergmann's rule. Previous research found that larger offspring were produced from sunny nests, but those larger offspring developed more slowly than shade offspring when reared in the lab. Since they developed more slowly, we'd expect that sun bees would have a lower metabolic rate than shade bees. However, Richards et al. 2020 found that sun bees actually had higher metabolic rates at high temperatures than shade bees. So the objective of my study was to investigate the mechanisms of the temperature size rule in mass provisioning bees, and I hope to clarify the seemingly contradictory observations of large, more slowly developing offspring that had higher metabolic rates than shade bees. And I hypothesized that provided maternal foraging effort is the same, so the size of the provisions that the offspring receives from their mother, provided that's the same, if sun bees are still smaller than shade bees, then the size difference is due to their metabolic responses to their developmental temperature. So C. calcarata is incredibly easy to experimentally manipulate and observe. Simply attach a raspberry cane to a stake, put the cane out in a sunny spot near some naturally growing raspberry or sumac in the spring, and you can attract nesting females who will start to dig a new nest. It looks like this with sawdust at the end. In the morning or in the evening, with the female inside, you can move the nest to a new experimental location. And they usually they move fairly well, provided that you do it early or late enough in the day. To test my hypothesis that body size differences are caused by metabolic responses to temperature, I use those methods to trap nest C. calcarata females and randomly assign them to either a sunny or a shady experimental site. I measured the nest and air temperatures in the sun and shade daily between 2 and 3 p.m. I observed foraging mothers and their foraging activity, and I measured adult offspring body size, metabolic rate, and offspring developmental rate. So this figure is showing the variation in air and nest temperature at 2 p.m. on the y-axis over the developmental period of C. calcarata from June to August on the x-axis. Unsurprisingly, I found that it was hotter in the sun, in the orange lines here, than it was in the shade, which is the blue lines. It was also hotter inside the sunny nest than it was outside. So the dotted lines are the internal nest temperature and the solid lines are the external air temperature. While the air and nest temperatures in the shade were about the same, so you can see that the blue lines are overlapping. And this suggests that maternal nest site does affect her offspring's developmental temperature. So this figure is showing the distribution of foraging flight activity by treatment group. So flight duration is on the x-axis, and the number of trips observed of that flight duration are on the y-axis. Both groups have the same right skewed shape, but on the right-hand side, the box plot shows that mothers from sun and shade foraged for similar durations, with duration of flight in minutes on the y-axis and the treatment group on the x-axis. And this suggests that mothers are not making any adjustments to their foraging behavior based on their nesting location. This figure shows offspring head width on the y-axis in millimeters and treatment group on the x-axis. And what this shows is that that bees born in the sun were smaller than bees born in the shade. And this confirms that microclimate does indeed affect offspring body size in C. calcarata. Figure here is showing the carbon dioxide burst frequency of sun and shade adults using discontinuous gas exchange at 25 degrees Celsius. So here on the right-hand side is just a representative photo of the metabolic rate trace with the peaks show as CO2 release over time. The first and last peaks are when I open the chamber to put the bee in or take the bee out. So the burst frequency is the number of CO2 bursts per minute. And over here on the left side, we found that bees from the sun released CO2 less frequently over the trial, suggesting that they have a lower metabolic rate than shade bees at 25 degrees Celsius. And finally, we compared the critical thermal maximum of sun and shade bees. And then this figure, CT max is on the y-axis and head width is on the x-axis. Sun bees had higher CT maxes than shade bees and are therefore more heat tolerant. This, in addition to the results from earlier, the Richards et al. 2020 paper, suggests that sun bees have a higher, that Richards et al. 2020 found that sun bees have a higher metabolic rate at 40 degrees. These results together suggest that sun bees perform better at higher temperatures than shade bees. And they may indicate that they are producing heat shock proteins in response to the high temperatures of their nest environment. So heat shock proteins are protective proteins that help cells function at high temperatures. But they're metabolically costly to produce because they take energy away from growth and development of the organism, and they divert it towards cellular maintenance. And this reduction in energy available for growth and development could be leading to the reduced body size in sun bees. So thermal compensation could be the mechanism responsible for the temperature size rule in mass provisioning bees. This research confirmed that sunny nests are hotter than shady nests, suggesting maternal choice affects offspring microclimate. I found that mothers do not adjust their foraging behavior in response to nesting microclimate, suggesting they don't compensate for the expected smaller size of their offspring in sunny nests. Richards et al. 2020, previous results of higher metabolism at 40 degrees Celsius in sun bees, coupled with what I found, the higher CT max and lower metabolic rate at 25 degrees Celsius in sun bees, suggests that they have a higher thermal tolerance and possibly a shifted thermal breadth in comparison to shade bees. So C. calcarata follows the temperature size rule, and in conclusion, the mechanism could be thermal compensation. I'd like to thank you for listening today. Thank my supervisor, Dr. Miriam H, for guidance during the project. My lab mates with help with fieldwork, Alex, Lily, and Lyndon for fieldwork assistance, and for Nora for helping me ID my specimens. Thank you.